DNA is a very powerful tool for a forensic scientist, so the ability to collect DNA from a crime scene is valuable. We will begin discussing DNA as evidence by doing a quick review of the structure and function of DNA. Some of you may have to challenge yourselves to remember this content from biology class. DNA stands for deoxyribonucleic acid. DNA is the molecule that stores and helps to transmit genetic information. DNA is tightly packaged into chromosomes which never leave the confines of the nucleus. This is a quick DNA timeline highlighting major events in DNA technology as they relate to forensic science. We will focus on a couple of these events, but it is worth noting that DNA evidence was first used in 1985. The FBI began using DNA evidence in 1988, and the federal DNA database called CODIS began in 1998. James Watson and Francis Crick received most of the credit for determining the structure of DNA back in 1953. But it was an X-ray photograph taken by Rosalind Franklin that helped the duo develop the final structure. The function and biological importance of DNA was also determined in the mid-1900s, and this led to an explosive advancement in cellular biology. Once DNA was determined to be the molecule that made each individual unique, it was only a matter of time before forensic scientists took advantage. Sir Alec Jeffreys developed the first DNA profiling technique that could be used to match DNA from a crime scene to the DNA of the victim or suspect. We will discuss this technique in more detail at a later time. Jeffreys used his technique to help exonerate an innocent man, Richard Buckland, accused of the rape and murder of 2 young women. Then he used the technique to convict the actual criminal. The Colin Pitchfork case is the first case that used DNA evidence to exonerate an individual and convict the actual perpetrator. Let's switch gears and discuss the structure of DNA. DNA is double-stranded, and the 2 strands coil together to make the double helix. DNA is a polymer, which means it is composed of multiple repeating units called monomers. The monomers of DNA are nucleotides. Each nucleotide is composed of a sugar molecule, deoxyribose, a phosphate group, PO4, and one of 4 nitrogen bases, adenine, thymine, guanine, or cytosine, or A, T, G, and C for short. The backbone of the double helix is on the outside. It is composed of the deoxyribose and the phosphate groups of the nucleotides. The nitrogen bases are arranged on the inside of the DNA double helix in what would seem to be a random order. However, the order of the nitrogen bases is nearly the same in all humans, 99.9% the same. Here is a diagram of DNA in the form of a double helix. You can see the repeating sugar-phosphate backbone and how the nitrogen bases form the rungs in the middle of the helix. The nitrogen bases form hydrogen bonds with each other, and it is these interactions that hold the double helix together. You will also notice that certain nitrogen bases are always paired together, adenine with thymine, guanine with cytosine. So A pairs with T and G pairs with C. This is why in a sample of DNA, the amount of adenine is always equal to the amount of thymine, and the amount of guanine is always equal to the amount of cytosine. For example, if a DNA sample contained 20% adenine, then it would also contain 20% thymine, because those 2 pair together. Which means the remaining 60% of the DNA sample is composed of guanine and cytosine. So the sample would contain 30% guanine and 30% cytosine, because those 2 bases pair together. DNA is packaged into chromosomes. Let's stretch that biology memory a little. Remember, human body cells have 23 pairs of chromosomes, which means 46 chromosomes in each cell. The chromosomes are paired because upon conception, a person gets 23 chromosomes, or half of their DNA, from mom, and 23 chromosomes, the other half of their DNA, from dad. One pair of chromosomes is the sex chromosomes, X and Y. Females have 2 X chromosomes, and males have an X and a Y chromosome. As we have briefly discussed, there are 2 sources of DNA in your cells, Nuclear DNA is the molecule we think about when studying DNA. Nuclear DNA is found in the nucleus of the cell. This is the type of DNA that forensic scientists seek because it serves as individual evidence. Mitochondrial DNA is found in the powerhouse of the cell, the mitochondria. And instead of being packaged as chromosomes, it exists as a circular DNA molecule. Mitochondrial DNA comes from your mother. A sperm cell has mitochondria, but only in the tail of the sperm cell. Upon fertilization, the head of the sperm enters the egg cell, but the tail is removed. Therefore, none of your father's mitochondrial DNA is transferred during fertilization. Forensic scientists can use mitochondrial DNA as class evidence to link a DNA sample to a specific family. But the process of mtDNA analysis is more time-consuming and more costly than analysis of nuclear DNA. The function of nuclear DNA is to store and transmit genetic information. Genetic information is stored in genes. Genes are sections of the DNA that code for specific proteins. These proteins have numerous functions in your cells and throughout your body. Any cell that contains a nucleus will contain DNA. Most body cells contain a nucleus. Hair roots, semen, cheek cells and saliva, teeth, and bones are included in this list. Cheek cells have an abundance of nuclear DNA, which is probably why a mouth swab is so valuable to a forensic scientist. When it comes to blood, only white blood cells have a nuclei. Red blood cells lack nuclei, therefore they have no nuclear DNA. The DNA molecules that make up each chromosome are ridiculously long. Now, the majority of the DNA molecule is non-coding, which means it doesn't code for a protein. That is illustrated here in this image. Here are a couple of coding regions, which means these regions include instructions for making a protein. But as you can see, most of the DNA does not code for a protein. These non-coding regions contain sequences of the bases A, T, G, and C that repeat over and over. As you will see, it is variations in these regions that are used in forensics. When DNA evidence is found at a crime scene, it is important to collect and preserve the sample correctly. This includes using disposable gloves and collection instruments. When collecting the sample, avoid talking, sneezing, coughing, and touching the evidence directly. Wet samples need to be air dried before being placed into paper containers. DNA evidence should be kept cool and dry during transport and should be stored in the freezer. Here is a list of methods that can be used to collect DNA evidence. Smear slides involve placing a blood sample onto a glass slide, sliding a second slide back into the drop, and using that top slide to smear the blood sample across the bottom slide. When blood is found on clothing, CSI collect and bag the entire article of clothing. Sometimes a cloth may be used to soak up excess blood. Any dried blood found on, say, an article of furniture would result in the entire piece of furniture being taken to the lab. Dried blood from a surface that cannot be transported to the lab is carefully scraped into an appropriate container. The DNA left at a crime scene could be affected by a variety of environmental factors, such as heat, sunlight, moisture, mold, and bacteria. Because of this, not all DNA evidence will be suitable to create a DNA profile. Even with a DNA profile, you can only determine the individual was at the scene. The DNA itself will not explain when an individual was at the crime scene or for how long he or she was there. If enough DNA evidence is collected, the DNA can be used to create a DNA profile. Remember that only about 0.1% of human DNA is different from one person to the next. So it is this small percentage of DNA that is used to create a DNA profile like the ones pictured here. We will learn how to create and read these DNA profiles during the course of this unit. DNA profiles are then stored in DNA databases. The State DNA Index System, or SDIS, contains DNA profiles from local forensics labs and the State Forensics Lab. The SDIS feeds into the National DNA Index System, or NDIS, which is obviously a national DNA database. In 1998, The FBA created the Combined DNA Index System, or CODIS, which allows labs throughout the country to access DNA profiles. CODIS is actually the software used to run the comparisons of DNA profiles. The database does not include any personal information, save for the birth date of a missing person. That was a lot of information I just threw at you. So let's take some time to process that. Until next time.